Okay, so today what we're going to be doing is we're actually going to be making a foldable. So you should have gotten some sheets of paper, and essentially this is what it's going to look like. So the two pieces of cardstock that you were given are going to turn into the top piece and the bottom piece. And then you've got 1, 2, 3, 4, 5, 6, 7, 8, 9 additional pieces of paper that you're going to kind of put together, and you're going to fold in this way, and then you're going to staple on the top. So I've got mitosis versus meiosis. And if you want to pause here, and you can actually write the different labels here on the tabs, that would be great. So we've got number of cells made, number of chromosomes, number of divisions, crossing over, question mark, cell description, function, and then on the left side we put prophase, metaphase, anaphase, telophase, and then prophase 1 and prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, and telophase 1, telophase 2 on the other side. Because what we're going to be doing is on these different pages we're going to be comparing the two. So on the first one, which is the number of cells made, what you're going to do is on one side you're going to write mitosis, and the other side you're going to write meiosis. Under mitosis we have 2 cells, and under meiosis we have 4 cells. Under tab 2, number of chromosomes, for mitosis we are diploid, so we are 2n. Now that can be any number, depends on the type of organism you're talking about. Whereas in meiosis the cells end up haploid, or n. For tab 3, the number of divisions, we have 1 division in mitosis, whereas we have 2 divisions that occur in meiosis. Crossing over, mitosis, we have no crossing over, but in meiosis we can have crossing over taking place, and that's because of the formation of the tetrads during prophase 1. The cell description, for mitosis we have identical cells with the same number of chromosomes and the same type of chromosomes. These are found in body cells, or this process happens in body cells. In meiosis, the cells are different. Each cell gets 1 chromosome from the homologous pair. So each cell is either going to get your chromosome that you got from mom or the chromosome you got from dad for that particular chromosome. The selection is random, and we end up with gametes or sex cells. So this is the process that takes place in their production. For tab 6, the function. In mitosis, it's growth and repair, as well as asexual reproduction. Remember that we do have a disorder. Cancer is uncontrolled mitosis. And its job is to maintain the chromosome number as we go through the cell divisions. With meiosis, it is sexual reproduction, and that's because our gametes fuse during fertilization to form a diploid, what we call zygote, and its job is to cut the chromosome number in half. All right, so now what we're going to do is go through and kind of compare the different different stages, but the ones that have the same name. So prophase of mitosis versus prophase 1 and prophase 2 of meiosis. So in mitosis, we get no pairing of homologous chromosomes, the spindle fibers form, and the chromatin coils into chromosomes. The cells are diploid, or 2n. With prophase 1, the DNA does replicate. The homologous chromosomes pair. That's the process of synapsis. Crossing over occurs, and the tetrads form. Again, the cells are diploid. Here, the chromosomes are double-stranded. In prophase 2, we don't replicate the DNA before this. The chromosomes are just moving to the center, and the cell is haploid, but the chromosomes are still double-stranded. In metaphase 1 versus metaphase 2, okay, in metaphase, in mitosis, the centrioles move to the poles, and the chromosomes line up with their centromeres on the equator. In metaphase 1 of meiosis, the homologous pairs line up across the equator from each other. The cells are diploid, and the chromosomes are double-stranded. In metaphase 2, the chromosomes line up with their centromeres on the equator, so it's very similar to that in mitosis, but here the cell is haploid, but the chromosomes are still double-stranded. In anaphase 1, an anaphase, With mitosis, the sister chromatids pull apart and separate to the poles, whereas in anaphase 1, the homologous pairs separate to the poles. The cell again is diploid, and the chromosomes are double-stranded. In anaphase 2, we get the sister chromatids separating to the poles, whereas the cell is diploid, and the chromosomes now are single-stranded. So I went from haploid to diploid, but I also went from double-stranded to single-stranded. The last tab, the back page, is your telophase versus telophase 1 and telophase 2. So in mitosis, we get 2 cells with single-stranded chromosomes, and it is diploid. In telophase 1, I end up with 2 cells with double-stranded chromosomes that are haploid. In telophase 2 of mitosis, I end up with 4 cells total at the end, single-stranded chromosomes, and I am haploid. Okay, so this is kind of a good way of comparing not only mitosis and meiosis, but even what's happening within some of the phases of meiosis, how they are different from each other.